Why your brain is physically rotting, and you can't stop it. The doctor dimmed the light and said the scans didn't explain the screams. Sleep had vanished weeks ago. Pills didn't help. He was sweating through sheets, whispering to a clock that wouldn't move. Then came the hallucinations, faces in the ceiling, a fire no one else felt. His heart wouldn't slow. His blood pressure spiked. He forgot his son's name. Within months, holes would pepper his brain like a sponge. There's no cure. Nobody survives. It's not alive. It has no DNA or RNA. It laughs at bleach, outlives scalpels, and spreads by shape alone. And it might already be inside you. We're going to show you true stories where ordinary moments, dinners, surgeries, a family's bedtime, turned into countdowns. We'll move fast, but we'll keep people first. You'll see why a protein shape can hijack a brain, what fails in hospitals, and where real risk actually lives, not the movie version. Here's the path. First, a family curse that ends in sleepless death. Fatal Familial Insomnia. Then we rewind to a Papua New Guinean epidemic called Kuru and the painful way the world learned how it spread. We'll hit the gamble that almost broke a scientist's career. The prion, an infectious protein that survives radiation meant to shred genomes. We'll map how a bad fold becomes a template, how it multiplies like gossip, and why it sometimes stops at a species border and sometimes doesn't. Keep a question in your head. How can nothing alive still spread? We'll walk into kitchens, farms, and operating rooms where micrograms matter. We'll meet deer that shed the problem into the soil and families who wait by the phone for test results. We'll look at rare medical transmissions and what hospitals change because of them. Then we'll face the hardest truth. Sometimes it starts with no exposure at all, just aging biology throwing a bad shape. We'll widen the lens to Alzheimer's and Parkinson's, where prion-like spread is real, but not the same as infection. Finally, we'll put tools on the table. Stronger sterilization, new diagnostics like RT-QuiC, smarter rules, and clear actions that protect you without panic. Along the way, we'll keep the humor dry and the facts sharp. The night sleep stopped. Fatal Familial Insomnia. He started counting ceiling tiles because counting sheep was a joke now. 2, 4, 7. He lost the thread, stared into the dark, and listened to his own heart hammer like a stuck metronome. Work said it was stress. A friend mailed lavender oil. His wife Maria watched the bedside clock and learned how long an hour can stretch when nobody in the room can sleep. By month 2, he'd lost weight without trying. His hands trembled pouring coffee. He laughed at nothing, abrupt, hollow, then snapped at his daughter for asking about homework. He apologized, cried, and then couldn't remember why he cried. The family doctor ordered a sleep study. Polysomnography told the quiet part out loud. Almost no deep sleep. Stage N3, the restoration phase, barely showed. REM flickered and fell apart. The text circled a flat line where dreams should be. A neurologist pulled up the scans. The thalamus, traffic control for the brain, the switchboard that helps sleep flip on and off, looked off, not ruined, but thinned. Autonomic storms followed at night. Sweat like rain, pulse racing, blood pressure climbing. He'd wake shaking, heart sprinting toward a finish line no one else saw. Sleeping pills dulled the edges and made him slower in the daytime, but the night stayed bright and merciless. Maria found a name online and hated it on sight. Fatal Familial Insomnia. It read like a dare. She learned there's a gene, PRNP, that encodes the prion protein, PRP. A specific mutation, D178N, can turn that protein into trouble, but only in the wrong context. At codon 129 in the same gene, a person can carry methionine or valine. When D178N meets methionine on 129, sleep erodes. When it pairs with valine, a different disease shows up. The same mutation, 2 outcomes, and either way, the end is hard. They sat in a genetic counselor's office, fingers knotted. One test could tell them if their daughter carried a 50-50 coin flip. Knowing wouldn't change treatment. There isn't one, but it would change how she saw every tired night for the rest of her life. They didn't decide that day. They ate sandwiches they couldn't taste and walked the block without talking. Meanwhile, his sleep shrank to minutes. He'd doze in a chair, eyes fluttering, body jerking awake like he was falling. Hallucinations joined, shadows that pressed on his chest, voices that arranged themselves into warnings he couldn't quite hear. The thalamus kept losing ground. This wasn't burnout, it was a circuit breaking. The family drew up schedules: who sat with him at midnight, who fielded work calls he couldn't answer, who hid the car keys when his judgment slipped. One night, Maria found him bargaining softly with his nightstand clock. "Just 5 minutes," he whispered. He offered memories in trade, like a person carrying valuables out of a burning house. She told him it wasn't his fault. He nodded, then asked where they were. In the clinic, a specialist explained prions without drama. The prion protein is normal. Every brain makes it. In fatal familial insomnia, that protein can misfold into a shape that persuades other copies to match it. A bad origami teaching a class, and it grades itself. The thalamus, packed with the normal version, gets seeded by the misfolded form. Clumps form, then fibers, then more seeds as fragments break off. The result looks like sponges under a microscope, holes where firing neurons used to be. There's no microbe to kill. Antibiotics and antivirals have nobody to hit. Sleep wasn't just absent, it was war. The body ran hot because the autonomic nervous system lost its governor. The mind unraveled because night stopped repairing days. He started wearing sunglasses indoors. Light felt harsh. Friends visited and watched him search for simple words like they were buried in sand. He gripped their hands and the grip would soften mid-sentence, attention sliding away. Genetic testing came back. D178N with methionine at 129. The counselor didn't say fate. She said options, advanced directives, hospice choices, research donation if they wanted to turn the end into a beginning for someone else. The family chose to name the days. They found a last good Sunday afternoon and ate ice cream in the park in winter coats, laughing at the cold as if it were a prankster they could shoo away. At home, Maria learned to time his storms. The sweating at 2 a.m., the shivers at 3, the brief window at 4 when chamomile tea was acceptable because it felt like doing something. A nurse taught her to watch for aspiration, to tilt his chin just so when he drank. Their daughter drew a sign for the bedroom door. Quiet, Dad's night. They kept notes, micro stories. The day he remembered the dog's name and forgot his own birthday. The morning he asked to hear their wedding song 3 times and then asked what the song was. Each entry was both a knife and a ribbon. They didn't fight the term insomnia anymore. They used it like people use a weather forecast. Less to argue with, more to prepare around. The specialist didn't sell false hope. He explained that even though prions spread by shape instead of genes, the logic is relentless. The seed converts to native form, the fiber grows, it breaks, it seeds again. You can't sleep past it. You can only make the room softer, the air warmer, the choice is clearer. They made one more. To record his voice on a day when sentences still held. He said his daughter's name slowly. He told Maria she'd rescued him every day, even the ones he couldn't see. To understand why sleep could vanish and never come back, and why a protein could hold a family hostage without being alive, we need to go back to the first human mystery that taught the world how a shape can spread. 3 decades earlier, in a mountain village half a world away, people started shaking, laughing, and falling. The answer wasn't in a virus or in a bacterium. It was in a ritual of love and in a protein that refused to die. Stay with this thread. The missing piece is about to flip how you think biology works. The Cannibal's Curse. Kuru in the 1940s. 3 decades earlier, high on the Okapa Plateau in Papua New Guinea, grief wore a public face. Women wailed, men cut their hair, and the South Four people practiced a mortuary rite meant to keep the dead close. Transumption. The careful consumption of relatives so their strength would live on in kin. It wasn't cruelty, it was devotion. Then something started to happen in kitchens and gardens where mourning turned to meals. Hands began to tremble, feet lost the ground, laughter burst out at the wrong times and wouldn't stop. Children and women died first, and whole hamlets felt hollowed out. The stakes were human, immediate, and lopsided. The rites centered in the household, and so did the risk. Women prepared the bodies, handled brains and viscera, and fed children. The disease had an eerie arc. Months of stumbling and shaking, odd giggles that masked fear, then swallowing problems, bedbound days, and silence. The elders said ancestors were angry, outsiders whispered about curses, but funerals kept coming and small graves kept appearing. 2 patrol officers wrote reports that reached physicians. In 1957, a young doctor named Carlton Gajdusek arrived with notebooks and questions, and he stayed. He watched, listened, and documented the clinical picture: ataxia, tremors, bursts of inappropriate laughter, then progressive loss of speech and movement. He tracked who fell ill and where they lived. Meanwhile, anthropologists like Shirley Lindenbaum and Robert Glass mapped social networks, kinship, and the routes grief took through daily life. The pattern made a terrible sense. The practice of honoring the dead was also transmitting something deadly. But what? Bacteria and viruses were the usual suspects. Samples shipped out of the mountains kept coming back without answers. Microscopes didn't show a germ. Culture plates didn't bloom. Still, the disease marched on. To test whether an infectious agent existed at all, Gajdusek did a brutal thing science sometimes requires. He took brain tissue from people who died of Kuru and inoculated chimpanzees. Then he waited. Years. That delay was its own message. This wasn't a quick pathogen. When the chimps developed a similar neurological decline after long incubation, it proved transmission. Kuru wasn't a curse. It was an infectious encephalopathy. The finding didn't end the epidemic. The community did. Colonial authorities had already pressured the 4 to stop transumption. Missionaries called it sinful, administrators illegal. As the practice faded in the late 1950s and early 1960s, new childhood cases dried up, but the long tail of incubation meant adults kept getting sick for decades. The last documented case appeared in 2009, a ghost echo from meals shared in the early 1960s. That timeline taught the world prions could hide in a body for years before showing their hand. What nobody knew at the time was the nature of the agent itself. Gajdusek earned a Nobel for demonstrating transmissibility, but not chemistry. Meanwhile, others were puzzling over a sheep disease called scrapie, with similarly weird resistance to UV and ionizing radiation, types of energy that rip through DNA and RNA. If radiation barely dented the agent, maybe the blueprint wasn't nucleic acid. The Kuru story, mapped onto household practice and incubation data, became a human-scale clue in a larger scientific riddle. Ethics ran alongside the science. The language of cannibalism landed like a slur. The 4 preferred transumption, a precise word for a ritual of love and obligation. Researchers learned, slowly, to listen to how the community named itself and grieved. Consent and partnership improved over time. The people who'd already lost so much weren't specimens. They were partners in figuring out what had happened and how to prevent it elsewhere. Back in the villages, the disease's signature moments became micro-stories families told with a hard kind of fondness. A mother who'd once carried sweet potatoes up a ridge without pausing could no longer trust her feet on a flat path. A boy's laughter at a cousin's joke turned shaky, then uncontrollable, then thin. The day a favorite song couldn't summon a dance step marked the point. Everyone feared. Decline had started its final run. Meanwhile, something else was happening in labs an ocean away. The chimp experiment had opened the door. Now scientists pushed into the room and started turning over furniture. If not a virus, then what replicated? In scrapie studies, Stanley Prusiner enriched infectivity in fractions dominated by a protease-resistant protein, PRP2730, and noted that UV and X-ray doses that should have wrecked genetic material didn't stop transmission. Different strains acted different while carrying no obvious genome. The evidence pointed to a shape, not a sequence. For the 4, the immediate impact of ending transumption was concrete. Fewer funerals, more children growing into parents. For the rest of the world, the impact was conceptual. Kuru proved humans weren't exceptions to a class of diseases already known in animals. It drew a line from a kitchen hearth in Okapa to abattoirs in Britain decades later, when bovine spongiform encephalopathy, mad cow disease, would show how industrial shortcuts could feed a prion back into a herd. It warned hospitals that tools used on brain tissue might carry more than stains if cleaned by ordinary rules. In the aftermath, scientists returned to the mountains not to reopen old wounds, but to close loops. No one born after the practice ended developed Kuru. That simple, devastating fact locked the transmission story. It also framed a new fear. If a cultural practice could move a misfolded protein through a community, what other roots existed in our connected world? Here's where the tone shifts. To understand why radiation didn't stop the agent, and why bleach struggled later, we follow the data into the moment. A scientist named a thing nobody wanted to believe in. The next chapter isn't about kitchens or chimps. It's about a word, prion, and a wager that a protein shape could act like a pathogen. The protein that cheats death, Prusiner's gamble. While that was happening in the mountains, a young neurologist in San Francisco was staring at data that didn't add up. Stanley Prusiner read the Kuru papers and then fell down the scrapey rabbit hole. Sheep with brains that looked like sponges, agents that shrugged off treatments designed to smash genomes. He kept coming back to a strange radiobiology result from Tikva Alper. UV at 250 nanometers and heavy ionizing radiation. Things that crippled DNA and RNA, barely touched infectivity. If a genome wasn't getting hit, maybe there wasn't one to hit. This was the moment everything changed for him. The central dogma said information flows from nucleic acids to proteins. If you want something to replicate, you need a code. Proteins were effectors, not genome. Proposing otherwise was career carbide. Colleagues warned him, "Don't say it out loud," he said it anyway. He coined a word, prion, for proteinaceous infectious particle, and he made a sharp claim in 1982. The essential agent of scrapey is protein. No hidden virus, no bizarre mini-chromosome, just a host protein misfolded into a conformation that can recruit more of itself. The stakes were real. Grant reviewers rolled their eyes. Some journals bounced the papers. In the lab, failures piled up, preparations lost activity, controls misbehaved. But one result wouldn't die. When they fractionated infected brain and chewed away nucleic acids, infectivity remained. When they degraded protein, infectivity fell. They enriched a core fragment, PRP2730, partially resistant to proteases, enriched for infectivity. The gene they mapped, PRNP, lived in the host, not in some phantom pathogen. It was like pulling the mask off the villain and finding your own face underneath. Meanwhile, radiation and UV controls kept reinforcing the heresy. Dose curves that would have vaporized a viral genome left titer stubbornly high. People argued maybe the agent was shielded by a protein coat that protected a tiny nucleic acid. Prusiner's team countered with independent inactivation methods, hydroxylamine, nucleases, intercalators that damaged nucleic acids without touching proteins. Infectivity didn't care. But when denaturants and proteases mauled protein structure, it dropped. Shape, not sequence, seemed to carry the instructions. Here's where it gets crazy without getting mystical. Strains, different clinical phenotypes and incubation times, popped up even when the agent supposedly lacked a genome. How do you get stable strains if there's no genetic variation? The answer, he argued, lived in conformation. A single amino acid sequence can adopt multiple stable misfolded states, each templating its own architecture. Think of steel paper clips bent into different jigs. Each jig can stamp more to match itself. No letters change, but the fold encodes behavior. The ridicule turned personal. He got nicknames, some not printable. A senior voice told him to stop disgracing virology. But other datasets began to line up. Inherited human prion diseases mapped to mutations in PRNP, like the D178N you just met in Fatal Familial Insomnia. In animals, scrapey and then bovine spongiform encephalopathy pointed to host genetics and exposure, not a foreign genome. The protein-only hypothesis didn't just fit, it predicted. Action in the lab meant an ugly truth. This material was hard to kill. Protease resistance wasn't absolute, but it was robust. Formaldehyde fixation and routine autoclaving, the backbone of hospital sterilization, left infectivity lurking. That resistance would later haunt operating rooms, but in the 1980s it mostly fueled skepticism. If it's protein, why doesn't it melt like other proteins? The answer was amyloid, a dense, beta sheet, rich structure that packs into fibrils, physically shielding bonds and aligning backbones so tightly that enzymes and heat struggle to get in. Not immortal, just stubborn. What nobody knew at the time was how deep the family tree would grow. Yeast prions, discovered later, showed that self-templating protein conformations were a broader biological trick, sometimes even beneficial. The human cost kept the focus sharp. Sporadic cases with no clear exposure, familial cases with cruel Mendelian edges, iatrogenic clusters from pituitary hormones or dura mater grafts. The variety made more sense once you accepted that the same protein could misfold into distinct templates, cross-seed in some contexts, and crash species barriers when conformations happened to match. In the aftermath of his gamble, the field reorganized around mechanisms. The story pivoted from what genome to what shape, and then to how does that shape spread. Funding thawed, methods improved. New assays began reading out seeding activity instead of hunting imaginary nucleic acids. The Nobel Committee eventually called, not to bless a personality, but to mark a conceptual rewire. Information can live in structure. This decision would haunt and help medicine. Haunt because tools and tissues once considered safe needed new rules. Help because once you frame disease as a templating process, you can imagine breaking it in two ways. Stop seeds from forming or stop seeds from teaching. To see how that lesson works inside a brain, we have to watch a bad fold recruit a good one. Shape as code, how misfolding spreads. To see the code, start with the normal version. PRP, in its everyday form, call it PRPC, sits on neuron surfaces like a polite neighbor. It's flexible, mostly alpha helical, and constantly chatting with the cell's quality control crew. Then a seed arrives. Pre-C-S-key, the same amino acids bent into a different architecture, rich in beta sheets. They meet. The seed offers a handshake that isn't a handshake. Contact flips a region, then another. The native protein matches the intruder's pose, and the pose is the message. One conversion doesn't wreck a brain, but the new convert sticks to the seed, extending it into a protofibril. Add more converts and the fiber grows. Beta sheets stack like pages in a damp book. Friction, cellular motion, and enzymes nick the fiber. It breaks. Now you don't have one seed, you have dozens. Each fragment carries the same misfolded template. That's the moment the graph curves upward. Bad origami teaching a class, and it grades itself. Cells aren't passive. They deploy chaperones that refold proteins, proteasomes that chew mistakes, lysosomes that digest junk. Early on, this proteostasis network keeps up. But amyloid fibrils resist proteases, and the fragments dodge through membranes. Meanwhile, the normal PRP is replenished because the gene keeps making it. The misfolded fraction climbs, tips a threshold, and the repair crew falls behind. Neurons don't explode. They fade, lose synapses, and leave holes like sponge marks under a microscope. Here's what nobody saw at first. The strain lives in the fold. PRP with the same sequence can lock into different beta sheet patterns, each with a distinct surface and energy profile. Those surfaces select which incoming PRP molecules can join. That's why clinical pictures differ, and incubation times vary, even without nucleic acids. Conformation is the cookbook, not the letters. But there's a catch, the species barrier. PE sequences differ between species by a handful of amino acids. Those differences shift the shape space of what's easy to template. A sheep-derived seed meets human PRPC, and sometimes the handshake just doesn't fit. Conversion is slow or never happens. Dose and route matter too. A mouthful is not a neurosurgical smear. A high dose can brute force a low probability fit. Direct inoculation into brain leaps past digestive defenses and hits neurons where PRP is dense. Meanwhile, something else is happening along axons. Neurons traffic cargo forward and back. Misfolded seeds catch a ride in vesicles or skim along membrane contact points. Seeding appears in one brain region, then shows up in connected areas in patterns that match wiring maps. It's not a marching army. It's gossip along a phone tree, moving by contact and convenience. You can watch the code in a tube now. In RT-QuIC and PMCA assays, you take a trace amount of seed, often from cerebrospinal fluid, add a flood of recombinant PRP, and agitate. Conversion starts slow, then surges, then plateaus when substrate runs out. Thioflavin dyes light up the beta sheets. The lag time and slope are fingerprints of seeding efficiency. No genomes, just structure, training structure. An early flag that guides care and infection control. Conformation even explains why bleach fails at ordinary strengths. Beta sheets pack tightly, burying vulnerable bonds. Surfaces adhere to steel, especially when dried, turning instrument crevices into seed banks. That's why protocols call for concentrated sodium hydroxide or hypochlorite, plus extended autoclaving at higher temperatures. Enough to denature and hydrolyze, not just sanitize. Hospitals learned this the hard way when standard sterilization left infectivity behind on tools used in neuro cases. Viral hook. The thing you trust to be clean can carry a memory you can't see. Now imagine a single seed landing in a brain where PRNP carries a destabilizing mutation. The energy barrier to misfolding is lower, the native fold breathes wider, and the handshake gets easier. That's familial disease. In sporadic cases, a rare thermal fluctuation or oxidative nick might flip a molecule for a moment. Most flips refold or get cleared. Very rarely, one lines up just right with a neighboring copy in templates. From there, the kinetics, not intent, decide the future. Dose and time carve the path. A small exposure might smolder for years, converting slowly until the fragment cloud reaches critical mass. A massive dose, like contaminated pituitary extracts once given to children, shrinks the fuse. Root sneaks in again. Oral exposures face proteases, stomach acid, and epithelial barriers. Peripherally, seeds may replicate on follicular dendritic cells before nerving in. Intracerebral exposure bypasses all that. Same code, different delivery. Not all misfolded forms talk to each other. Cross-seeding depends on surface compatibility. Some prion strains barely convert across species. Others hop with tweaks. That variability keeps epidemiologists up at night. It also gives drug hunters a lever. Block the key surface, and you block the message. Conformation-specific antibodies, peptidomimetics that cap fibril ends, or small molecules that favor the native fold. Each bets on interrupting the handshake. What nobody knew at the time of the first Kuru transmissions was that soils could archive the code. In chronic wasting disease, prion-laden saliva and carcasses load the environment. Minerals like montmorillonite clay bind PRPSC and paradoxically boost its uptake in animals. The template persists longer than anyone wanted to believe, and every hoof print is a chance meeting with the seed. The impact of all this isn't theory, it's a timer. Once seeding kinetics pass a point, symptoms accelerate because every new fragment creates more conversion sites. That's why patients look stable then slide. It's also why early detection matters. Fewer seeds mean more time for any intervention to matter, if and when we have one that does. So the code is a fold. The fold is the message. The message spreads by touch. The next question is brutal and practical. How do those touches reach us in real life? On plates, on steel, and in dirt. The edges of exposure, food, tools, and dirt. While that handshake spreads inside a brain, the real-world question is simple. Where does the first hand come from? Start with dinner plates. In the late 1980s and early 1990s, British cattle developed a strange sway and fear, their version of spongy brains. Bovine spongiform encephalopathy wasn't a folk curse, it was an industrial shortcut. Rendered remains, including nervous tissue, were recycled into feed. One cow with seeds became 1,000 mouths. Grinding it all together erased the obvious line between sick and safe. When humans ate contaminated beef, a small number developed variant creutzfeldt-jakob disease. Not regular CJD. This one hit younger people and showed distinct patterns in the brain and on pathology slides. The route was probable. Oral exposure to tissues rich in prions, especially brain and spinal cord. Dose mattered. Millions consumed burgers, but the risk concentrated where the seeds lived. Feed bans and specified risk material rules, removing high-risk tissues from the food chain, dropped cases dramatically. The lesson wasn't panic, it was plumbing. Control what goes in at the start, and you don't have to guess at the end. Now switch rooms, operating theaters. Steel doesn't rot, but prions love it. Dried on a neurosurgical instrument, seeds adhere to microscopic scratches and cling through routine sterilization. An autoclave run that destroys bacteria leaves this template yawning. Hospitals learned this the worst way: iatrogenic transmissions. In the past, growth hormone extracts from pituitary glands collected from cadavers carried prions into children. Dura mater grafts sourced from donors did the same for adults. A handful of corneal transplants and neurosurgical instrument clusters added more sorrow. Each case forced a rewrite of the manual. Sterilization got asterisks. If you suspect high-risk tissue, you don't rely on the ordinary cycle. You hit instruments with concentrated sodium hydroxide or strong hypochlorite, then run extended high-temperature autoclave protocols. In some scenarios, you don't reuse at all. Single-use tools for specific brain and eye procedures. It's not that bleach never works, it's that the usual bottle and a quick wipe are theater, not chemistry. The real fix is a combination: denature, hydrolyze, and buy yourself a margin. Meanwhile, something else was happening outside the walls. Deer and elk were shedding prions into the open. Chronic wasting disease doesn't wait for a scalpel. It moves by saliva, urine, feces, and carcasses. Those fluids hit leaves, water, and especially soil. Certain clays can bind PRPSC and perversely make it more available to passing animals. The environment turns into a reservoir, not for days, but for seasons and years. A salt lick becomes a meeting place and a multiplier. Hunters adjusted. Some states and provinces posted maps with hot zones. Camps bought test kits or mailed lymph nodes to labs. Don't eat until it's negative turned into a rule you could tape to a cooler. The waiting by the phone wasn't theater either. It was a way to compress the unknown into a week instead of a lifetime. Agencies banned feeding and baiting that crowded animals, limited carcass transport across borders, and told processors to keep heads and spines out of grind piles. Not perfect, but better than shrugging at the woods. Back in the kitchen, the risk calculus narrowed. Commercial beef after feed bans, with specified risk materials removed, tested, and tracked by lot, is not the same situation as the BSE peak. Steak isn't magic armor, but reputable supply chains and regulations drive known risks down hard. Wild game in CWD country is different. You don't guess, you test. If positive, you don't eat. High heat doesn't. Rescue a bad result. Prions aren't bacteria waiting to be pasteurized. Hospitals move beyond sweeping policies into checklists. If a patient might have CJD, path labs change blades and traps. Endoscopes that touched lymphoid-rich tissues get flagged. Instruments exposed to suspected prion tissue are quarantined until diagnosis clarifies. Blood services added donor deferrals for people who spent time in high-risk regions during the BSE era. The point wasn't to stigmatize, it was to stop shape from riding a bus it never should have boarded. There's an uncomfortable middle ground too. Dental tools, ENT scopes, and procedures that touch tissues with lower PRP expression. The guidelines weigh probability and practicality. You don't autoclave the whole hospital in sodium hydroxide, but you do segment instrument sets, track patients, and escalate decontamination when the index of suspicion rises. It's adult medicine, layered defenses, not talismans. Soil, steel, and steak share one ugly trait. Tiny amounts can matter, but that truth cuts two ways. Tiny amounts plus the wrong route and tissue can light a fuse. Tiny amounts handled by smart systems and barriers become background noise. The story of exposure is less about a single villain and more about choreography. What touches what, how long, at what dose, and whether we bother to break the chain. Here's the dry humor beach you can use when your friend brags about cleaning. The stain that laughs at bleach and then invites friends. Only stops laughing when you change the whole party. Different chemicals, longer heat, different tools, and a guest list that excludes brain and spine from the buffet. The next question walks out of the lab and into the tree line. If the woods are loading themselves with seeds and animals are moving them mile by mile, what does that mean for families who hunt, for ecosystems that lean on deer, and for the uneasy border between species? Zombie deer, chronic wasting disease, CWD. Walk into a frosty November morning in Wisconsin and you'll see the stakes emblaze orange. Families set up camp, brew coffee on tailgates, and tell the same jokes their grandparents told. Then someone mentions the map, colored dots spreading across counties, and the laughter thins. Chronic wasting disease is here. It doesn't make deer snarl or crave brains. It makes them fade, ribs showing, heads drooping, saliva threading from the mouth. A hunter tracks a buck for hours, takes a clean shot, and then wonders if he just harvested a question mark. The action isn't a single outbreak, it's a slow creep. First identified in captive mule deer in Colorado in the late 196s, CWD moved into wild herds and then across state lines like spilled ink. Today it's confirmed in multiple states and Canadian provinces, plus parts of Scandinavia. The transmission web is messy and efficient. Infected deer shed prions in saliva, urine, and feces long before they look sick. They nuzzle, lick, share scrapes, crowd salt licks, and everything they touch becomes a potential handoff. Carcasses magnify it. Scavengers drag parts. Soils inherit seeds. Science chased the spread with better eyes. RT-QuIC and PMCA, lab assays that amplify seeding, made it possible to detect tiny amounts of prion activity in tissues and fluids. Lymph nodes from the head and neck, tonsils, and even feces can carry enough seed to light up a tube. Field teams collect samples from hunter-killed deer. Labs shake tubes and read the curve within hours to days. A strong signal doesn't just say infected, it says infectious potential, and that changes what families do with a freezer full of meat. Meanwhile, researchers press the species barrier from the lab side. Rodents engineered with cervid PRP catch CWD readily. Mice with human PRP resist far more. Studies in macaques split nerves. Some showed no disease after years of exposure. Others found subtle pathology or biochemical signals after oral or intracerebral dosing with CWD material. The takeaway isn't doom, it's uncertainty with teeth. There's a barrier, but it isn't a wall you bet dinner on. Agencies repeat the line that matters. Don't eat meat from a positive animal. Period. Curiosity gap. If a barrier slows spread, what happens when dose and route change? Policy tried to slow the dance. Feeding and baiting bans aimed to stop deer from clustering unnaturally. Carcass transport rules kept spinal cords and heads, the prion-rich parts, out of gas station dumpsters across borders. Check stations popped up like seasonal pop-up stores. Hunters learned a new ritual. Bag the retropharyngeal nodes, drop them at the tent, wait for a call. If the test is negative, you wrap and label. If it's positive, you don't take a chance. You don't feed it to pets. You dispose of it per rules that read more like biohazard than barbecue. The human beat is small and private. A father and daughter team drags a 10-point buck to the truck, high fives, then gets quiet when they fill out the sample card. They drive home, hang the deer in a cold garage, and check their phones every hour for 5 days. When the result pings negative, relief is loud. When it pings positive, the garage becomes a triage site. Gloves come out, knives get segregated, meat goes into bags they never thought they'd use for food. The daughter asks if they did something wrong. The father says no, they did everything right by checking. Meanwhile, the woods don't care about feelings. Prions ride out winters. Certain clays bind PSC and can even boost its uptake when deer root and graze. Plants may wick up particles on surfaces, making the interface between soil, leaf, and tongue a quiet conveyor. Land managers argue over culling versus targeted removals, over whether reducing density slows spread or just breaks traditions. Ranchers with captive herds face business-ending decisions when tests find a hot pen. Scientists push for nuance without murk. There's no evidence CWD transmits to humans through casual contact. Hunters who test and follow guidance dramatically cut their risk. Cooking doesn't fix contamination. Knives and grinders need segregating and cleaning with prion-aware protocols. Heads, spines, and offal shouldn't ride in coolers to cabins 2 states away. These aren't abstract rules. They're speed bumps that actually slow an epidemic measured in seasons, not days. The humor you cling to at camp gets drier. Somebody taps the bleach bottle and says it needs a meaner cousin. Somebody else jokes about gossiping molecules at the salt lick. The jokes aren't denial, they're armor for the waiting. What nobody knew when CWD first appeared in pens is how long the landscape can hold a memory. Now we do. It changes how we hunt, butcher, share meat, and talk to kids about why their deer needs a test before it becomes chilly. It also hands hospitals a mirror. If soils can keep a code, so can steel. The next chapter walks into operating rooms and asks a blunt question. When medicine forgets that shape can travel, who pays? When medicine forgets, iatrogenic transmission. Unknown to them, across town, a different kind of hunt was underway. Inside a hospital, on polished steel. A neurosurgeon finished a delicate biopsy, rinsed an instrument set, and sent it down to sterile processing. The trays came back gleaming. Days later, someone said the word nobody had put on the chart yet: CJD. A shape might have just taken a ride to another patient. Hook. How do you stop a memory that clings to metal? The stakes are trust and time. People walk into hospitals believing the tools are clean in the way that matters. With prions, clean needs an asterisk. In the 197s and 80s, children needing growth hormone got pituitary extracts pulled from cadavers. A few vials carried a hidden code. Years later, some of those kids, now adults, developed creutzfeldt-jakob disease. The dose had been tiny, the route was direct. The timeline felt cruel. Dura mater grafts did it too. Patches taken from deceased donors, treated but not prion-killed, were used to repair skull-based defects or cover the brain's surface. Some patients, years after routine surgeries, began to stumble, jerk, and forget. A handful of corneal transplants passed the shape through an eye, and rare clusters traced to neurosurgical instrument sets that looked spotless but held a template in their scratches. Each case forced hospitals to rewrite muscle memory. Here's the action, not the myth. Standard sterilization wrecks bacteria and most viruses. Autoclaves run at 121 degrees Celsius for 15 minutes, steam under pressure, and the bugs die. Prions aren't bugs die. Prions aren't bugs. Amyloid folds pack so tight that water and enzymes can't get teeth in. Dried onto steel, the seeds adhere like barnacles. A quick brush, routine detergent, and a normal autoclave cycle leave enough structure to matter. That's how a template moves from one skull to another. So the decon reality changed. When brain or spinal cord might be involved, biopsies, deep ENT cases near lymphoid tissue, pituitary and posterior eye work. You don't trust the usual. Protocols call for concentrated sodium hydroxide or high-strength sodium hypochlorite soaks, followed by extended autoclaving at higher temperatures and longer times. Some instruments don't survive that. That's a feature, not a bug. For certain high-risk steps, single-use tools are now the norm. Path labs swap blades and change traps before opening a suspected CJD case. Endoscopes that touched lymphoid-rich tissue get flagged and sequestered pending diagnosis. Meanwhile, what nobody knew at the time of the first iatrogenic reports is how much behavior needed to change on paper. Disclosure policies matured. If an instrument set might have been exposed before a diagnosis, infection prevention teams track where it went and who it touched. Patients get calls nobody wants to make or take. The counseling is blunt and careful. The absolute risk is low but not zero. There's no screening blood test, there's no post-exposure pill to swallow. The goal isn't panic, it's respect for uncertainty. Blood services made their own moves. People who lived for long stretches in places and times with high BSE exposure were deferred from donating. It wasn't a verdict on them, it was a gatekeeping choice to keep a shape off the highway. Tissue banks mapped donors more tightly. Dura and cornea sourcing shifted to systems that minimized unvetted material. Growth hormones switched from pooled human extracts to recombinant products. No more cadaver in a vial. Here's the humor beat that keeps you from grinding your teeth. The stain that laughs at bleach, and then invites friends, shuts up when bleach shows up with sodium hydroxide, extra heat, and a calendar. It's chemistry, not vibes. Inside operating rooms, micro-stories stack up. A scrub tech holds an instrument and asks the circulating nurse to check the case notes again. A pathologist pauses before cutting and swaps cassettes. A resident learning their first brain biopsy also learns a new alphabet of warnings taped to the wall. NaOH here. Do not reuse there. Quarantine tray 3 if prion suspected. None of this makes surgery scarier. It makes it smarter. Impact ripples out. National guidelines now spell out risk tiers by tissue. Brain, spinal cord, posterior eye, and pituitary at the top. Lymphoid-rich mucosa and dorsal root ganglia in the middle. Blood and most peripheral tissues far lower. Hospitals segment instrument inventories so a possible exposure doesn't contaminate the whole fleet. Documentation got less romantic and more exacting. Lot numbers, tray IDs, patient labels that follow a path like airline luggage with better ethics. And still, there's residual risk. Sporadic CJD can look like many things for weeks. A patient may enter the system labeled rapid dementia or rule out autoimmune encephalitis. Only later does an MRI pattern or a cerebrospinal fluid RT-QuIC result light up the prion path. That lag is where iatrogenic stories begin. The fix isn't clairvoyance, it's default caution for certain tissues and procedures, plus fast pivots when suspicion rises. The counseling lands in a human place. A surgeon sits with a family and explains that an instrument possibly touched high-risk tissue before the diagnosis was known. The chance of harm is very small. The hospital will monitor. There's nothing to do today but live your life. The family nods, asks hard questions, and then goes home to a dinner that tastes different. Trust bends, but doesn't have to break when systems tell the truth. What this chapter proves is simple and uncomfortable. Shape travels on steel if we let it. The countermeasure is choreography, chemistry, heat, segregation, and paperwork that reads like a shield. And remember the woods? Those same principles. Don't move heads and spines. Don't mix toolkits. Don't guess.
Sporadic CJD and aging proteomes.
3 days after the hospital scare, a different story walked in without any exposure at all. No transplant, no surgery, no deer camp. A retired school principal started telling the same story twice at dinner. A month later, she forgot the route to a grocery store she'd driven for 20 years. Her daughter said stress. Her son said grief. The ER said possible stroke. The MRI said something else. Diffusion changes in the cortical ribbon, bright where brain should be quiet. The shape had arrived without an invitation. This is the villain nobody can point to. Sporadic creutzfeldt-jakob disease shows up in about 1 person per million each year worldwide. There's no known trigger you can circle in red ink. It's a roll of molecular dice in a body where, with age, the table tilts. Proteins spend their lives folding, refolding, and getting cleared. That balancing act, proteostasis, runs on chaperones, proteasomes, lysosomes, and autophagy. With age, the crew tires. Oxidative stress scuffs surfaces. Heat shock responses lag. The same quality control system that once turned misfolds into confetti now lets a few slide by. Somewhere in that latency, a single PRP molecule may flip into a rare conformation. On most days, it snaps back or gets eaten by the cleanup squad. On a bad day, it fits against a neighbor just right and teaches the wrong pose. From there, it's the same kinetics you've seen. Growth, fragmentation, more teaching. No deer, no donor, no instrument, just time and chance pushing a molecule across an energy ridge. Clinically, the slope is steep. Families talk about a good week that already feels off, then a month where speech shortens, attention frays, and myoclonus, those sudden jerks, startle everyone at the table. Gait goes shaky, vision blurs, sleep grows strange. A neurologist asks for tests that sound like alphabet soup. CEG, MRI, CSF, RT-QuIC. The EEG sometimes shows periodic sharp wave complexes, spiky patterns that whisper prion. The MRI's diffusion-weighted images can flash bright in the cortex or deep nuclei, mapping the spread like heat marks on a wall. Cerebrospinal fluid, dripped through an assay that reads seeding RT-QuIC, lights up with growing fluorescence as PRP in the tube learns the wrong fold. It's not a horoscope, it's kinetics on camera. Meanwhile, the differential diagnosis is a crowd. Autoimmune encephalitis can sprint, viral encephalitis can confuse, toxic metabolic crashes can mimic. Doctors throw antibodies and antivirals when those lanes seem likely, because they're the lanes with exits. But when the pattern holds, rapid dementia, myoclonus, a kinetic mutism creeping in, CJD comes to the front. A brain biopsy could confirm, but often, with imaging and RT-QuIC, it's not needed. Families already have the answer they didn't want. Stakes land with paperwork and kitchens. There's no cure. Median survival is measured in months. Caregivers quit jobs. Siblings fly in. Someone Googles at 2 a.m. and stares at the words spongiform change and pictures holes. The better picture is synapses dimming, networks failing, a city losing power block by block. Hospice becomes a plan you make while still setting coffee for the morning. Advanced directives jump from theoretical to urgent. Durable power of attorney, do not resuscitate orders, who knows passwords, life admin as love language. Here's a micro story that makes the science feel human. The principal had a favorite sweater she wore on exam days. Blue, soft at the elbows. In week 6, she reached for it and asked whose it was. In week 8, her son put it on her shoulders and she smiled anyway, without recognition, because kindness still made sense to a nervous system that couldn't name it.
The disease takes nouns first, then verbs, then the quiet glue between people.
Families learn to meet presence without words. What nobody knew in the 199s, but we lean on now, is how much earlier we can read seeding. RT-QuIC turns suspicion into evidence without a scalpel. A few drops of CSF, hours of shaking in a plate, and the curve climbs if seeds are there. Not perfect, but precise enough to change how we talk. It also sharpened infection control when cases present as unknown encephalopathy, labs flag, instruments quarantine, and the iatrogenic stories you just heard get rarer. Genetics still hangs in the background. PRNP codon 129, methionine or valine, doesn't just dictate familial outcomes, it shapes sporadic risk and phenotype too. Methionine homozygotes are overrepresented in certain SZJ subtypes, and incubation in variant disease tracks that background. It's not destiny, it's a tilt. Aging proteomes slide faster on some slopes than others. Treatment trials come and go with headlines that fade. Anti-aggregation compounds, conformation-specific antibodies, copper-binding ideas, chaperone boosters. None have moved the clinic needle yet. The timelines are crueler than our patients. Still,
Early detection tools matter.
if a therapy that slows seeding ever clears the bar, catching the disease when seeds are few will buy time you can feel. Impact ripples into law and money. Families scramble to secure disability benefits, protect savings, and guard against fraud targeting vulnerable elders. Social workers become navigators. Palliative teams manage symptoms. Myoclonus with clonazepam. Sleep with careful routines. Anxiety with presence more than pills. Swallowing assessments prevent aspiration. Beds get rails, rugs go away. The home becomes a controlled landscape, not a museum of the past. Meaning is the last hard chapter here. Sporadic CJD reminds us that randomness is a player in our lives, not a footnote. You can do everything right and still meet a bad fold. The response isn't superstition, it's intention. Advance directives while you're well. Conversations that scare you a little now, so they hurt less later. Donations to brain banks that turn endings into data futures can use. Dignity isn't a cure, but it's a technology. It makes the final months less brutal for everyone in the room. And then there's presence. Families mark last good days. They plan small rituals, a favorite song at lunch, a brief walk at sunset, a call with an old friend on speaker so the voice can run across the room. The disease writes its timeline. People write around it, leaving notes for themselves in the margins so they remember the person before the template took their words. Beyond prions. Prion-like spread in Alzheimer's and Parkinson's. While families faced the speed of CJD, another set of families were living a slower heartbreak. A grandmother forgot recipes she'd made for 40 years. A former engineer's hands began to tremble, then curl, then refuse the buttons on his shirt. Doctors said Alzheimer's, Parkinsons, familiar names with unfamiliar mechanics. Here's the careful line. These aren't infections like Kuru or variant CJD, but inside the brain, something eerily similar happens. A misfolded shape teaches neighbors the wrong pose. Start with Alzheimer's. Two proteins drive the discussion, beta amyloid and tau. Beta amyloid, a chopped fragment from a larger precursor, aggregates into plaques outside neurons. Tau, a microtubule stabilizing protein, misfolds inside neurons into tangles. The mystery wasn't that they accumulated, it was how the pattern spread. Brock and Brock mapped tangles marching from the entorhinal cortex through limbic circuits and into neocortex, stage by stage, as if following wiring diagrams. That map looked less like random rot and more like a route.
In lab dishes and animal models, seeds of misfolded tau or amyloid injected into one region trigger pathology that appears later in connected regions. It's not replication in the infectious sense, it's templating, seeding mechanics echoing prions without the human-to-human epidemiology. The seed lowers the energy barrier for the next copy to match its shape. Neurons ship cargo along axons, vesicles bud, fuse, and leak. Exosomes and synapses act like ports. The misfolded core catches rides the cell built for work, not sabotage. Now slide to Parkinson's. The key protein, alpha-synuclein, normally helps with synaptic vesicles. Misfolded, it assembles into Lewy bodies. Clinicians noticed a strange prodrome: constipation, loss of smell, REM behavior disorder, acting out dreams, years before tremor. Brack staging again traced a path, this time from the olfactory bulb, and gut up the vagus into midbrain nuclei where dopamine neurons live. In animals, alpha-synuclein seeds placed in gut walls later show up in brainstem, cut certain nerves, and the spread slows. The phone tree metaphor holds. Here's what nobody knew in the early Alzheimer's era. You can read this templating in tubes, like with prions. RT-QuIC variants now detect alpha-synuclein seeding activity in cerebrospinal fluid, and in research settings, even in skin or olfactory mucosa. For tau, seeding assays exist too, capturing the lag-then-surge curve that betrays templating. The word prion-like lives here carefully to describe the mechanism, seed and template, not the epidemiology. There's no credible evidence that casual contact spreads Alzheimer's or Parkinson's between people. The spread is within one brain circuits. Boundaries matter. Classic prion diseases cause transmissible spongiform encephalopathies with well-documented iatrogenic and dietary routes in rare contexts. Alzheimer's and Parkinson's are not like that in everyday life. Still, scientists pay attention to edge cases. Old Dura-Mater graphs, the very kind linked to iatrogenic CJD, have produced patients with beta-amyloid deposits at surgical sites years later. That doesn't mean their dementia traveled in. It means amyloid can seed in tissue when placed there. It's a caution about instruments and tissues, not a cue to fear handshakes. The stakes of the prion-like frame are huge. If misfolded tau or alpha-synuclein spreads by templating along circuits, interventions can target the handoff. Antibodies that bind the misfolded species might soak up extracellular seeds. Small molecules could cap fibril ends, so growth stalls. Peptidomimetics might wedge into the beta sheet zipper and jam it. Chaperone inducers could boost the cells, refold and destroy Kuru before the avalanche starts. And because staging maps predict which regions go next, clinicians can time treatments to circuit vulnerability, not just symptoms. Action in trials has been messy. Anti-amyloid antibodies, aducanumab, lecanemab, lower plaque burden, and nudge clinical endpoints, with side effects like ARIA, brain edema, and microbleeds. The wins are modest, the debate's loud, but the core idea stands. Intercept the seed, slow the spread. Tau-targeting therapies aim closer to the neuron's dying breath. Some try vaccines, others go after kinases that hyperphosphorylate tau and make it aggregable. In Parkinson's, anti-alpha-synuclein antibodies and small molecule aggregation blockers have entered and left phases with mixed results. The field is learning the oldest lesson. Biology is stubborn. Kinetics favor the early mover. Meanwhile, diagnostics are stepping forward. CSF RT-QuIC for alpha-synuclein is approaching clinical use to distinguish Parkinson's and dementia with Lewy bodies from lookalikes. Seed amplification assays for tau variants promise to sort Alzheimer's from frontotemporal tauopathies, a split that guides families through different futures. Blood tests for phosphorylated tau forms offer easier screening, though they read downstream signals, not seeding itself. Pair those with PET ligands for amyloid and tau, and doctors can see the map while the patient still remembers the way home. Humor helps keep the gears turning. It's gossip, but molecular, and it travels by neuron. That line sticks because it's true enough to remember and careful enough to avoid panic. The gossip doesn't jump rooms by air. It follows private lines. Shut down the rumor mill early, and the story dies out. One more human beat. A son notices his father's voice getting quieter. At dinner, napkins become origami cranes. The hands won't finish. A doctor explains staging without stealing hope, talks sleep and exercise and community. Tools that don't cure, but steady the days. The family signs up for a study that collects spinal fluid and scans every year. Data piles up, and somewhere, a lab's curve reaches its plateau on a Tuesday, and a postdoc grins because a cap on a fibril finally held. What this chapter adds to our story is a bridge.
Prions taught us that shape can act like code. Alzheimer's and Parkinson's show that code can move within one person's brain without turning into an epidemic. The same mechanics, tuned by different proteins and contexts, write different fates. And if shape can carry illness, then shape can also carry therapy, design folds that interrupt the lesson mid-sentence. Countermeasures, from bleach-proof to breakable. After all that, the obvious question lands hard. If the enemy is a fold, how do you break it? Not with vibes, with chemistry, heat, separation, and timing. Start with the old rules rewritten. Routine sterilization kills life, but prions aren't alive. So hospitals built prion-aware playbooks. If an instrument touches high-risk tissue, brain, spinal cord, posterior eye, you don't send it through a normal wash and steam. You soak it in concentrated sodium hydroxide or high-strength sodium hypochlorite, then autoclave at higher temperatures for longer runs. That double hit denatures and hydrolyzes the stubborn beta sheets. Some tools warp. Good. A ruined wrong-ewer beats a second patient. But the real move is to keep steel out of the line of fire. Single-use kits show up where risk clusters, pituitary work, deep ENT near lymphoid clusters, posterior eye procedures. Path labs isolate blades and change vacuum traps before opening suspected prion tissue. Instrument tracking trails every tray like a detective story, where it's been, what it touched, and whether it needs quarantine. It's not paranoia, it's choreography that turns a template's stickiness against itself. Diagnostics got sharper too. RT-QuIC and PMCA let labs detect seeding in a few drops of cerebrospinal fluid, nasal brushings in research settings, even lymphoid tissue for animal surveillance. That means faster pivots. A patient shows rapid dementia. MRI hints at prion, CSF RT-QuIC lights up, and infection prevention flips protocols same day. The earlier the flag, the fewer instruments wander, the fewer calls anyone has to make months later. Speed isn't a cure. It's a wall you can actually build. Meanwhile, food systems learned hard lessons and kept them. Feed bans stopped recycled, nervous tissue from looping back into herds. Specified risk material rules keep brain, spinal cord, and certain gut parts out of the human food chain. Traceability tags lots, so a bad node doesn't take down a season. Hunters in CWD country fold testing into their calendar. If a deer is positive, it doesn't get eaten or fed to pets. Knives and grinders get segregated. Heads and spines don't ride to new zip codes. Not perfect, but precision beats panic every time. Now to the bench, where shape fights shape. Conformation-specific antibodies try to spot the exposed surfaces unique to PRPSC and either flag it for clearance or block it from converting fresh substrate. Some designs aim to bind fibril ends, where growth happens, and cap them, turning jagged saws into dull stubs. Peptidomimetics mimic a slice of the beta sheet zipper, lodge themselves in the seam, and jam assembly. Small molecules nudge the equilibrium back toward the native fold or destabilize the misfold enough that the cell's cleanup crew can finally bite. Proteostasis boosters come at the problem sideways. If the repair shop's understaffed, hire. Chaperone upregulators, autophagy activators, and proteasome enhancers aim to clear the junk sooner, lowering the odds a stray seed survives long enough to teach. The risk is blunt. Push too hard, and you stress cells in a new way. Trials need to show that tuning the cleanup crew helps the whole symphony, not just the brass section. Then there's the nuclear option. Edit the gene. Animals engineered without ProRP don't get prion disease from exposure. No substrate, no spread. In theory, silencing PRNP in adult humans could turn an ongoing fire into embers. In practice, you need targeted delivery to the brain, safety over decades, and an answer for what normal PRP does that you'd be removing. Gene therapies for monogenic prion families sit on distant horizons. They're signals, not solutions yet. Drug timelines deserve honesty. The kinetics are unforgiving. By the time symptoms show, seeds aren't single digits, they're cities. That's why people chase prevention in high-risk families, catch early, act early, or push for screening tools that spot seeding in pre-symptomatic windows. If you can shave the reproduction number of seeds below 1, the curve bends. That's a big if. Trials with anti-aggregation compounds and antibodies haven't delivered clinic-changing wins yet. But every failure tightens the map of what not to try next, and that's progress with fingerprints. Lifestyle becomes a quiet part of countermeasures, not because kale blocks prions, but because behavior blocks routes. Hunters test and follow disposal rules. Surgeons disclose and document. Families push for advanced directives so decisions don't get made in chaos. Blood services keep donor deferrals that look outdated to some and essential to others. Each small guardrail lowers the odds a seed finds steel, steak, or a spinal canal. Humor keeps morale from rusting. The stain that laughs at bleach and then invites friends goes quiet when bleach shows up with sodium hydroxide, extra heat, and a schedule. Translation: use the right tool long enough in the right order. One more human beat. A sterile processing tech tapes a new card above the sink. Prion protocol checklist. Boxes to tick, times to write. A hunter teaches a teenager how to find the retropharyngeal nodes and why waiting for a test is part of respect for the animal. A nurse practitioner adds, talk advanced directives to a routine physical for patients over 50. None of these feel heroic, they feel boring. But boring saves brains. So countermeasures aren't magic, they're layers. Chemistry plus time, heat plus segregation. Early flags plus honest calls. Science that treats the shape as code and then writes a smarter. How likely is it for you? After all that fear, here's the math you can live with. For most people, prion disease isn't a lurking shadow. It's a lightning strike. Sporadic CJD hits roughly 1 person per million per year worldwide. That's rarer than being struck by lightning twice. Familial cases tied to PRNP mutations make up about 10 to 15% of human prion disease. Iatrogenic transmissions, through old pituitary extracts, historical duragraphs, or contaminated neurosurgical tools, are now extraordinary outliers because systems learned and changed. Variant CJD, the human echo of mad cow, rose with the BSE crisis and fell after feed bans and specified risk material rules kicked in. In many countries, yearly deaths from VCJD have dropped to single digits or none for years. Blood donor deferrals for people who spent time in high-risk places during that era remain in force. They're guardrails, not verdicts. In short, the wave passed and surveillance kept the shoreline watched. Region matters for wildlife. If you hunt where chronic wasting disease circulates, you're living with a different equation. Still low, but not zero unless you test. Agencies publish CWD maps by county and provide testing routes for retropharyngeal lymph nodes. When the sample is negative, the chili pot can come out. When it's positive, you don't eat it. You don't feed it to pets. You follow disposal guidance that treats the carcass like a code you don't want to copy. Clarity helps more than bravado. You don't catch human prion disease from casual contact, shared air, hugs, or living in the same house. Cooking temperatures that make chicken safe don't neutralize prions, but reputable commercial beef, processed under modern regulations and with specified risk materials removed, carries vanishingly small risk. The infamous exposures came from brains and spinal cords slipping into products before rules hardened. Those tissues are now removed by design. Hospitals aren't minefields. If your surgery doesn't involve high-risk tissues, brain, spinal cord, posterior eye, pituitary, your exposure risk is effectively nil. When those tissues might be involved, prion-aware protocols, instrument tracking, and when indicated, single-use tools cut risk to the bone. If a possible exposure ever happens, you'll hear about it. The call is awful. The absolute risk is still tiny. So what should you actually do? Here's the short, actionable ledger. If you hunt in a CWD area, test every deer or elk before eating. Wear gloves when field dressing. Avoid cutting through brain or spinal tissue. Keep knives and grinders for wild game separate from kitchen gear. Don't move heads and spines across borders. Follow disposal rules. Buy meat from reputable sources that follow feed bans and specified risk material rules. If you travel, the same rule applies. Trusted supply chains beat rumors. In healthcare, tell your team about any prior neurosurgeries, duragraphs, pituitary hormone treatments from the 197's 80s, or corneal transplants. Most people don't have these histories. If you do, transparency helps infection prevention do its job. If a loved one develops rapid dementia, myoclonus, or other red flags, ask whether MRI diffusion, EEG, and CSF RT-QuIC are appropriate. Early identification won't cure, but it sharpens care and protects others in clinical settings. If prion disease runs in your family, meet a genetic counselor before testing. Know what you'll do with the answer. Consider research registries and brain donation programs that turn grief into data. Basic life planning isn't morbid, it's merciful. Advanced directives, durable power of attorney, and simple checklists spare families from crisis decisions. whether the diagnosis is prion disease, stroke, or anything else. Perspective keeps anxiety from squatting in your kitchen. Millions handled venison last season, only a fraction hunted in CWD areas, an even smaller fraction harvested positives. The proportion who ignored guidance is smaller still. Hospitals perform millions of procedures. Prion-related events are headlines precisely because they're rare. The world isn't sterile, but it's not reckless either. Here's your humor with a use-by date. Hug friends. Don't lick elk carcasses. Ever. Wash your hands because life is messy, not because prions care. Trust systems that earned it and keep asking good questions so they don't get lazy. The risk ledger ends where living begins. Respect the code. Break the chains you control. Then go make dinner without letting a rare fold rent space in your head. The last good day. Care, grief, and memory. While everyone else argues molecules, a family watches the clock. They circle a date on the calendar and call it the last good day, knowing the label might be wrong but needing something to hold. Breakfast still tastes like breakfast. A favorite song still pulls a hum. The neurologist's words hang in the air. No cure. Months, not years. Plan now. Care starts with choices, not heroics. Hospice isn't surrender, it's a trade. Less hospital, more home. Less poking, more presence. A nurse walks in with quiet shoes and a notebook. She talks pain scales, constipation from meds, how to keep nights gentler. Myoclonus gets a small dose of clonazepam. Anxiety meets voices and steady hands before pills. Sleep becomes a ritual. Lights low, phone down, familiar blanket. The same story at the same time, every night. Legal paperwork stops being a chore and turns into armor. Advanced directives, durable power of attorney, healthcare proxy, names on lines that spare sons and spouses from guessing during a storm. Someone sets up automatic bill pay. Someone else lists passwords and prints a one-page cheat sheet for doctors. Diagnosis, meds, allergies, who to call. It's not morbid, it's merciful. The house changes shape. Rugs roll up. A shower chair appears. Swallowing assessments guide meals. Thicker liquids, slow sips, small bites. The blender becomes a friend. A speech therapist coaches safe swallowing and ways to keep conversation going with fewer words and more cues. Pictures on the fridge, big print notes, yes/no cards for tired afternoons. A physical therapist teaches transfers that protect backs and dignity. Bed rails go up. The drinking glass gets swapped for one with a lid and a friendly straw. Community steps in if you let it. A neighbor takes Tuesdays so a spouse can nap. A sister handles insurance calls and holds on the line until a person answers, not a maze. A faith group or book club moves meetings to the living room. Someone drops off soup and takes the trash without asking for permission. Respite hours keep caregivers from breaking. Burnout is real. So is the relief of a shower you don't rush. Grief doesn't wait for the end. It starts when the first wrong name lands or the first locked door becomes necessary. Families learn to grieve in fragments. A daughter keeps a notebook of tiny victories. Dad laughed at the dog. Mom held my hand during the thunder. We watched the sunset and she said, "Pretty." They mark last without saying the word out loud. The last drive, the last walk to the mailbox, the last cup of coffee shared while it's still hot. Rituals hold memory when memory can't hold itself. Morning becomes music time. One playlist, familiar and forgiving. After lunch, a photo album with sticky notes. Your sister, our first apartment, the lake. Even when names slip, feelings stay. The nervous system forgets nouns before it forgets comfort. A soft sweater, a favorite scent, a dog's warm weight at the feet. These anchor a day better than any calendar app. There's a path for giving back that doesn't ask the patient to fight a war they can't win. Research donation programs walk families through brain or tissue donation with respect. Consent forms arrive early, not at midnight on the worst day. A coordinator explains what happens, how anonymity works, how a last hard choice can turn into data that spares another family down the line. It's not for everyone. For some, it's a way to turn helplessness into action. Micro stories keep the room human. A grandson learns to paint nails and does a shaky but proud job in bright coral. A spouse tapes a note to the fridge. "We're safe. You're home. Coffee at 9. At 9:07, they laugh because the habit of punctuality still taps the glass. The TV volume creeps higher each week. Someone finally buys headphones and the neighbors thank them with cookies. Honest conversation beats myths. The nurse says what choking looks like and how to respond without panic. The doctor explains comfort feeding and why forcing calories can cause harm. The social worker names anticipatory grief and hands over a local support group schedule. People cry in front of each other and don't pretend it's dust in the room. The last good day isn't a photograph. It's a practice. You find it by making space for small joys, by trimming tasks that don't matter, by letting help in, and by saying the true things while there's time. I love you. You were a good dad. You made this house a home. The science told us how fast the slope is. The care tells us how to walk it. Slower, softer, together. The bigger lesson, fragility and responsibility. Pull back from microscopes and campfires, from operating rooms and kitchen tables, and the pattern snaps into view. Our systems break when speed outruns caution. On farms that recycled nervous tissue to cut costs, in hospitals that trusted routines built for bacteria, in labs that once dismissed a protein with no genome. Vigilance fatigue is real. The lesson is simple and demanding. Shape is code, and code rides habits. Policies that worked weren't magic. Feed bans starved BSE at its source. Specified risk materials were carved out of supply chains. Dura and pituitary sourcing changed. Growth hormone went recombinant. Blood services held firm on donor deferrals. Infection prevention teams wrote prion protocols that look fussy until you map a single mistake forward. Each move shortened the routes a fold could travel. The temptation now is always the same. Declare victory, loosen bolts, forget the math. Don't. Complacency isn't a cartoon villain. It's a calendar. New staff rotate in, old stories fade, budgets tighten, and checklists get streamlined. That's when a dried smear on steel becomes history repeating. Keeping guardrails up means building habits that outlast individuals. Instrument tracking baked into software. Prion flags embedded in electronic orders. Annual refreshers that aren't optional, game day drills that walk a suspected CJD case through a hospital without leaving a trail. On the land, the same ethic holds. Feeding and baiting bans save traditions by saving herds. Check stations and transport rules aren't red tape. They're riverbanks that keep spread from braiding into new watersheds. Hunters teaching teenagers to test before they cook are doing epidemiology with buck knives. State agencies posting hot zone maps and turnaround times give families back their weekends without rolling dice. Meanwhile, labs and surveillance net the quiet edges. RT-QuIC didn't just improve diagnosis. It made iatrogenic mistakes less likely by tightening timelines. Wildlife surveillance that uses lymph node seeding assays finds hotspots before stories do. The unglamorous part is funding. Surveillance only works if it's boringly consistent. When budgets wobble, the map goes fuzzy. When the map goes fuzzy, shape gets room to move. Personally, responsibility is smaller and closer. Ask the extra question at pre-op. What tissue, what instruments, what protocol? If the answer is crisp, everyone relaxes. If it's muddy, you just did quality improvement. In clinics, add advanced directive chats to routine visits the way you add vaccines. Quiet, regular, no drama. If your family carries PRNP mutations, meet genetic counselors early and plan on your timeline, not the diseases. Humor is armor when it points toward action. Keep the dry lines that stick. It's gossip, but molecular. The stain that laughs at bleach stops laughing with heat, NaOH, and time. Hug friends, don't lick elk. Those aren't jokes for their own sake, they're memory aids for rules that matter when nerves are high or sleep is short. Close the loops we opened. Sleep died one household at a time in fatal familial insomnia, and families learned to count minutes like coins. The four people changed a ritual and ended an epidemic, long before Western journals caught up. A stubborn neurologist bet his reputation that shape could be code. He was right, and hospitals had to follow. Deer taught us landscapes remember. Steel taught us policies forget unless we make forgetting hard. So build systems that assume drift and correct for it. Reward the boring audit. Celebrate the instrument tech who quarantines a tray on a hunch. Fund the field station that mails bad news to a hunter in time to make a different dinner. Support registries and brain banks that turn private grief into public knowledge. Curiosity isn't a hobby here, it's a safety device. Keep it lit, Everything about prions whispers the same sentence. We are fragile, therefore we are responsible. Because shapes carry memory, our choices do too. A fold you'll never see can erase a lifetime in months, but the habits you keep can stop that message from traveling. If you want the next mystery, how hologram physics bends reality without breaking it, watch the video waiting on your screen. Subscribe for more untold science that treats fear with facts, and share this with the hunter, nurse, or neighbor who needs the real guardrails, not myths.